Good morning, students. So, in the last session, we talked about the structure of the DNA. DNA, we found out, consists of nitrogen base, sugar, and phosphate. And that structure is called as a nucleotide. And the two DNA strands are polynucleotide strands. Each DNA has about 3.3 billion nucleotide pairs. 3.3 billion nucleotide pairs, structures of which are called as genes. And one gene has about 3,000 nucleotide pairs, and we have around 30,000 such genes in the human DNA. All genes are identical, made of sugars, phosphates, and nitrogen bases. The only difference between the genes is the sequence of the nitrogen base pairs. All genes have ATGC sequences which are different from each other, and only a permutation combination of four alphabets ATGC make all the genes become different from each other and form different proteins in our body. And differences between we all, including humans, chimpanzees, lions, tigers, earthworm, every living organism is made up of a DNA, and all the proteins that are formed in their body only differ with a permutation combination of four alphabets ATGC. An amazing phenomenon created by nature. This DNA keeps on replicating. How did the replication take place? When the first cell zygote divided to form two blastomeres, the DNA underwent replication to form two identical daughter DNAs. And both blastomeres have in them identical daughter DNAs. Proof kya hai iska? If you remember, if the two blastomeres separate, they will form identical twins. So the daughter DNAs are genetically the same. Can you imagine? We have replicated two DNAs, maternal and paternal, into four DNAs. And four DNAs have again become two DNAs, two DNAs. One maternal, one paternal, one maternal, one paternal, like we did in mitosis, in cell division. And DNA had undergone replication. I hope you remember, cell division, we had done that. DNA replicates in which phase? DNA replicates in the S phase of interphase. DNA replicates in the S phase of interphase. Let us find out how does this DNA replicate in the S phase of the interphase. How does DNA replication take place? Let's understand this beautiful concept today. The DNA replication takes place by a mechanism called as the semi-conservative method of DNA replication. Semi-conservative. When one mother DNA forms two daughter DNAs, each daughter DNA has in it one of the mother's original strand. So this is the mother's original strand, which is found in the daughter DNA. And these are new strands. So these are new strands. And thus, one mother DNA will form two daughter DNAs, each daughter DNA retaining one of the mother's original strand. This is the mother's original strand, and one is a new strand. So the dotted one, black, is your a new strand. And one mother DNA forms two daughter DNAs, each having original strand, new strand, original strand, and new strand. Hence, this is called as the semi-conservative method of replication. Semi-conservative half conservation method of replication. Semi-conservative method of replication. Where does this DNA replication take place? The replication takes place inside the nucleus. Of course, DNA is found in the nucleus, so replication takes place inside the nucleus. How does this replication take place? First, there is activation of nucleotides. There are four types of DNA nucleotides found in the nucleoplasm in the form of monophosphate. So we have deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, deoxycytidine monophosphate. And ये है न्यूक्लियस ये है न्यूक्लियस सेल you have adenine plus deoxyribose sugar. Deoxyribose sugar. At second carbon, what do we have? H. So it is not OH, it is H. So adenine plus deoxyribose sugar. H at the second carbon. And attached to it is one phosphate. So this is called as deoxyadenosine monophosphate. Similarly, we have deoxyguanosine monophosphate, or you have a single nitrogen base wala deoxythymidine monophosphate or deoxycytidine monophosphate. And these monophosphates are converted into triphosphates. They are activated into triphosphate like DATP, DGTP, DTTP, and DCTP using ATP in the presence of an enzyme called phosphorylase. So, ये है adenine, double ring. ये है ribose sugar which has at the second carbon OH. So this is adenosine. It's not deadenosine, it is adenosine. And there are three phosphates. Adenosine triphosphate. Now, an enzyme called phosphorylase will act on adenosine triphosphate, remove the two phosphates. Remove the two phosphates and give the two phosphates to adenosine, or rather, this is deoxyadenosine. Second carbon pe we have H. So this is deoxyadenosine, and deoxyadenosine and if you remember, this is monophosphate. So this was found in the nucleus. Deoxyadenosine monophosphate. So this is deoxyribose sugar. And phosphorylase removes two phosphates from adenosine triphosphates and converts it into adenosine monophosphate. Adenosine monophosphate. And ATP becomes AMP. And deoxyadenosine monophosphate के ऊपर phosphorylase supplies two phosphates. Two phosphates. So what does deoxyadenosine monophosphate now become? Deoxyadenosine triphosphate. Deoxyadenosine triphosphate. So what is present inside the nucleus? Adenosine, ADP. ATP, adenosine triphosphate. What does phosphorylase do? Removes two phosphates, and adenosine triphosphate becomes adenosine monophosphate and adenosine deoxyadenosine, which is DAMP, deoxyadenosine monophosphate के ऊपर phosphorylase supplies the 2-phosphate to become deoxyadenosine triphosphate. So once again, in the nucleus was found deoxyadenosine triphosphate, deoxyadenosine monophosphate, deoxyadenosine monophosphate, deoxyadenosine monophosphate था, and phosphorylase acted on ATP and adenosine triphosphate. ट्राइफॉस्फेट में से दो फॉस्फेट निकाल दिए, एडेनोसिन ट्राइफॉस्फेट में से दो फॉस्फेट फॉस्फोराइलेज ने निकाल दिए and give it to deoxyadenosine monophosphate, which will then become deoxyadenosine triphosphate and adenosine monophosphate. So once again, if you see this carefully, you have adenosine, this is adenosine, this is phosphate, phosphate, phosphate. This is adenosine triphosphate. ATP. And this is deoxyadenosine monophosphate. And phosphorylase enzyme removes two phosphates and gives it to deoxyadenosine monophosphate, which will then become deoxyadenosine triphosphate. And adenosine triphosphate becomes adenosine monophosphate. तो ये हो गया deoxyadenosine triphosphate, and this becomes adenosine monophosphate. So what did adenosine triphosphate become? Adenosine monophosphate. And what did deoxyadenosine monophosphate become? Deoxyadenosine triphosphate. Which enzyme removes the two phosphates from adenosine triphosphate and makes into deoxyadenosine triphosphate? Phosphorylase. Phosphorylase does that. Similarly, DGMP plus ATP becomes DGMP. DGTP, I'm sorry, DGTP plus AMP. So deoxyguanosine monophosphate plus adenosine triphosphate में से दो फॉस्फेट निकल जाते हैं and gets added to deoxyguanosine monophosphate, which becomes deoxyguanosine triphosphate, and adenosine triphosphate on losing to phosphate becomes adenosine monophosphate. So again, two phosphates are removed from ATP and given to deoxyguanosine monophosphate, which becomes deoxyguanosine triphosphate. Similarly, deoxycytidine monophosphate plus ATP. ATP loses two phosphate to become AMP, and two phosphates are added to deoxycytidine monophosphate to become deoxycytidine triphosphate plus adenosine monophosphate. Similarly, deoxythymidine monophosphate plus ATP loses two phosphates to become AMP, adenosine monophosphate, and the two phosphates are added to deoxythymidine monophosphate to become deoxythymidine triphosphate and 2 adenosine monophosphates. So all the monophosphates, DAMP, DGMP, DTMP, DCMP, are converted into DATP, DGTP, DTTP, DCTP. So monophosphate become triphosphate, which is a substrate used for it, ATP. And what does ATP do? Loses two phosphates. What does ATP become? AMP. Adenosine triphosphate becomes adenosine monophosphate. Who will do it, sir? The enzyme called phosphorylase. And what will deoxyadenosine monophosphate plus 2 phosphate become? Deoxyadenosine triphosphate. What will deoxyguanosine monophosphate plus 2 phosphate form? Deoxyguanosine triphosphate. What will deoxythymidine monophosphate with 2 phosphate form? Deoxythymidine triphosphate. What will deoxycytidine monophosphate form with 2 phosphate? Deoxycytidine triphosphate. So all the monophosphates have been converted into triphosphates, and that is called as activation of nucleotides. Which enzyme helps in activation of nucleotide? Phosphorylase enzyme. So ये न्यूक्लियस के अंदर डीएनए है एंड उसके अंदर अब डीएटीपी है, डीजीटीपी है, डीसीटीपी है एंड डीटीटीपी है, which is all present in the nucleus. So those are all the activated nucleotides. Then origin or initiation points. DNA replication begins at certain specific sites situated, which are called as origin points or initiation points. There are several origin points. So ये है DNA, and DNA replication begins at multiple points, and those multiple points are called as origin points. Origin points, which are also called as initiation points. तो ऐसे multiple points है DNA में, and at the origin points, DNA replication is broken by an enzyme called as incision enzyme or endonuclease by forming an incision nick, and it starts the breaking of the hydrogen bonds and unzipping. तो ये है डीएनए और ये है उसके origin points. ये है origin points. And origin points randomly associated anywhere at the DNA. At the origin point, incision is made, cuts are made with the help of incision enzyme, also called endonuclease. Endonuclease will cut the DNA. Which bonds will it break? DNA के कौन से bonds को तोड़ेगा? If you remember, DNAs are held together by phosphodiester bonds. So, incision enzyme or endonuclease will break the phosphodiester bonds. If you see the bonds carefully, it will break these bonds. It will break these bonds. तो ये bonds तोड़ेगा DNA at the origin point. At origin points, these bonds are broken. So which bonds are exactly broken? Which bonds are broken? If you remember, phosphodiester bonds. If you remember, these are phosphoester, 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 phosphoester. And incision enzyme breaks the phosphodiester bonds. And DNA के मल्टीपल फ्रैगमेंट्स मिल जाएंगे आपको, डीएनए के मल्टीपल फ्रैगमेंट्स मिल जाएंगे. सो द एंटायर डीएनए is broken by phosphodiester bonds. So endonuclease or insertion enzyme breaks DNA phosphodiester bonds and DNA के multiple fragments मिल जाएंगे आपको. DNA gets broken into multiple fragments. So ऐसे बहुत सारे fragments बन जाते हैं. Now unwinding of the DNA takes place after the activity of endonuclease. The two DNA strands are separated, are free from each other but not separated. Strands are free from each other but not separated. The separation of the strands is done by DNA unwinding protein called as helicase enzyme, also called as replication protein. तो DNA unwinding protein or helicase enzyme, also called replication protein, starts breaking the hydrogen bonds. तो ये है replication bonds or replication units are broken by DNA unwinding protein, also called helicase. And those helicase enzymes will break the hydrogen bonds. So if you picturize it, समझो आपके पास दो कॉटन थ्रेड्स है और दोनों कॉटन थ्रेड्स के बीच में आपने बहुत सारी मैचस्टिक्स लगा के रखी है, and you catch hold of that beautiful structure. So two cotton threads plus matchsticks. So it looks like a ladder. दो cotton threads with matchsticks है, it looks like a ladder. You catch hold of the uppermost matchstick and start rotating it. It will look like this. It will look like this, like the ladder is rotating. So this is your DNA. And you take a scissor and you cut the thread. So if you cut the thread, the entire ladder will become many pieces. उसके multiple pieces बन जाएंगे. And each piece के अंदर you take your fingers and start breaking the matchsticks. Start breaking the matchsticks. Now, after breaking the matchsticks, the two DNA threads which were coiled, rotate कर रहे थे और एक fragment के अंदर वो coils थे. अब matchstick तोड़ दोगे तो because of the centrifugal force, the two segments जिसमें matchsticks टूट गई है will separate. And now when you rotate, it will look like this. So once again, break, fragments, rotating, matchsticks broken. So now the fragment looks like this. यहाँ matchsticks है और जहां matchsticks टूट गई है, because of the centrifugal force, the threads separate and it looks like this. So what are the two cotton threads? DNA strands. What are the matchsticks in between? Hydrogen bonds. We have caught hold of it and rotating. What is this rotation called as the double helix? Then we take a scissor and break the cotton threads. What is a scissor? Endonuclease, incision enzyme. Which bonds does it break? Phosphodiester bonds. Phosphodiester bonds. Phosphodiester bonds. and we get multiple fragments. We get multiple fragments. And then with the help of fingers, we break the hydrogen bonds. With the help of fingers, we break the hydrogen bonds. Matchsticks. So who is the finger in this case? Helicase, also called replication protein. And as the hydrogen bonds are broken, matchsticks are broken, what will happen to the two DNA strands? The two DNA strands which were coiled now will uncoil. Now they will uncoil. So once again, who are the cotton threads? Sugar phosphate, sugar phosphate, sugar phosphate. DNA, backbone. What are the matchsticks? Hydrogen bonds. What is the rotation of that ladder called? DNA double helix. What is the scissor called? Endonuclease. What is breaking of the threads called? Breaking of phosphodiester bonds. What are the multiple fragments you get, sir? The fragments are the ladders which are still rotating. What are your fingers which break the matchsticks called? Endonuclease, also called rep protein. Which bonds has it broken? Matchsticks means hydrogen bonds. And the two DNA strands which are coiled will now uncoil. And that uncoiling is now looking like an inverted Y. Coiled, uncoiled. So this is uncoiled. This looks like an inverted Y. तो यहां अभी भी कॉइल है और यहां हाइड्रोजन बॉन्ड्स टूट चुके हैं. So two DNA strands which were coiled will now uncoil, and this will look like an inverted Y, which is called as the replication fork. What is that called as? The replication fork. Let us see it. So here we had DNA. ये कौन से बॉन्ड्स थे? फॉस्फोडाइस्टर बॉन्ड्स. समझो ऐसा था. And which were these bonds? Phosphodiester bonds. And we have broken the phosphodiester bond with the help of endonuclease. Then we started breaking the hydrogen bonds. Hydrogen bonds are broken with the help of helicase. And as hydrogen bonds break with the help of helicase, the two DNA strands will uncoil. And it looks like an inverted Y. Looks like an inverted Y, which is called as the replication fork. What is this fork called as? The replication fork. It looks like a fork. Ab fork se apple ga rahe ho. This is a fork, and this fork is called as the replication fork. Now, the two strands are separated from each other. Hydrogen bonds टूट चुके हैं, but the two strands may again come close to each other and hydrogen bonds वापस बन सकते हैं. तो दोनों strands को अपने हेलिकेस की मदद से सेपरेट तो किया, बट दोनों strands न्यूक्लियोप्लाज्म में घूमते घूमते अगर वापस क्लोज आ जाएंगे तो उनके नाइट्रोजन बेसिस के बीच में वापस हाइड्रोजन बॉन्ड्स बन जाएंगे. सो द टू strands, separated strands, are prevented from coiling again by SSBP, single strand DNA binding protein or helix destabilizing protein. तो सेपरेटेड स्ट्रैंड्स का वापस से कॉइलिंग कौन प्रिवेंट करेगा? एसएसबीपी, सिंगल स्ट्रैंड डीएनए बाइंडिंग प्रोटीन. तो एक बार दोनों स्ट्रैंड सेपरेट हो गए हैं और ये रेप्लिकेशन फॉर्क बन चुका है. तो रेप्लिकेशन फॉर्क में वापस दोनों स्ट्रैंड्स क्लोज आके उनके बीच में हाइड्रोजन बॉन्ड्स वापस नहीं बनने चाहिए, अदरवाइज रेप्लिकेशन नहीं होगा। तो दोनों स्ट्रैंड्स को पकड़ के रखा जाता है। Both strands are caught together, and these strands are held in their positions by an enzyme called as SSBP, single strand DNA binding protein. Single strand DNA binding protein will keep the two strands tightly in their positions. दोनों स्ट्रैंड्स को अलग अलग करके पकड़ के रखा जाता है with the help of single strand DNA binding protein. Now, once that is done, check it here. There is synthesis of new strand. Each separated strand now acts as a template or a mold for the synthesis of a complementary strand. So this strand is now called as a template or a mold. The old strand is called template or a mold, and उसके ऊपर कॉम्प्लीमेंट्री न्यू स्ट्रैंड बनता है। मोल्ड के ऊपर ये बन रहा है न्यू स्ट्रैंड। सो दिस इज द ओल्ड स्ट्रैंड and this one is the new strand. What is old strand called as? Template or mold, और उसके ऊपर बनता है new strand. The new strand formation takes place with the help of a small RNA molecule called as RNA primer molecule, with the help of an enzyme called RNA primase. तो ये है टेम्पलेट, और टेम्पलेट के ऊपर बनता है एक मॉलिक्यूल कॉल्ड एज आरएनए प्राइमर मॉलिक्यूल। तो आरएनए प्राइमर मॉलिक्यूल नाम का एक मॉलिक्यूल बनता है। That molecule is made with the help of an enzyme called as RNA primase. तो ये separated strands है, और separated strands पे बनता है molecules called RNA primer molecule, which is a RNA molecule. तो DNA replication में पहले क्या बनता है? RNA. तो ये है दोनों separated strands, और यहां बनता है आरएनए प्राइमर मॉलिक्यूल। तो ये है आपका टेम्पलेट, ये है टेम्पलेट, और यहां बनता है मॉलिक्यूल्स कॉल्ड आरएनए प्राइमर मॉलिक्यूल। एंड आरएनए प्राइमर मॉलिक्यूल्स आर मेड विद द हेल्प ऑफ एन एंजाइम कॉल्ड आरएनए प्राइमेज एंजाइम. Primase enzyme is made with the helps in the making of RNA primer molecule. The RNA primer molecule attracts complementary nucleotides from the nucleoplasm, and that takes place with the help of DNA polymerase enzyme. And DNA polymerase enzyme brings in new nucleotides. DNA primase enzyme, also called DNA polymerase enzyme, brings in new nucleotides. तो ध्यान से, ये है टेम्पलेट, टेम्पलेट के ऊपर आ गया आरएनए प्राइमर मॉलिक्यूल, यह है आरएनए प्राइमर मॉलिक्यूल। एंड आरएनए प्राइमर मॉलिक्यूल जो है, आरएनए प्राइमर मॉलिक्यूल जो है, रीड्स द टेम्पलेट, इट रीड्स द टेम्पलेट, और टेम्पलेट को स्टडी करने के बाद it attracts new nucleotides. It attracts new nucleotides and helps in the synthesis of the new strand. So new strand synthesis takes place with the help of RNA primer molecule. So once again, this is the template. This is the template, or the old strand, old strand, बीच में क्या बन गया? आरएनए प्राइमर मॉलिक्यूल. RNA primer molecule. And then DNA polymerase brings in new nucleotides to construct the new strand. तो ये कौन सा बन गया? New strand. So once again, how many structures are involved here? This is the template or old strand. बीच में क्या है? RNA primer molecule. That is in between. RNA primer molecule is in between. एंड उसके ऊपर ये क्या बन जाता है? न्यू स्ट्रैंड. सो वी हैव टेम्पलेट, वी हैव आरएनए प्राइमर मॉलिक्यूल एंड वी हैव न्यू स्ट्रैंड. वी हैव टेम्पलेट, दिस इज द टेम्पलेट. टेम्पलेट के ऊपर ये है आरएनए प्राइमर मॉलिक्यूल्स मेड विद द हेल्प ऑफ प्राइमेज एंजाइम, and then DNA polymerase brings in new nucleotides and constructs the new strand. So who constructs the new strand? The enzyme called DNA polymerase constructs the new strands. It brings in complementary activated nucleotide. If you remember, सबसे पहले फॉस्फोराइलेज एंजाइम की मदद से हमने न्यूक्लियोटाइड्स को एक्टिवेट किया था. न्यूक्लियोटाइड्स गॉट एक्टिवेटेड and nucleotides after activation were delivered by DNA polymerase. तो समझो, this is guanine and adenine. Okay, let me show this again. This is guanine, adenine, thymine, cytosine. बीच में है आरएनए प्राइमर मॉलिक्यूल. And DNA polymerase will bring in guanine का complementary deoxyadenosine triphosphate. Deoxyadenosine triphosphate जो एक्टिवेटेड था. एडेनाइन की सामने आ जाए, सॉरी, डीऑक्सीसाइटिडीन ट्राइफॉस्फेट. आई एम सो सॉरी. GATC are on the template. And this is RNA primer molecule. बीच में. And guanine को पढ़ के आएगा डीऑक्सी adenosine triphosphate. Sorry, deoxycytidine triphosphate. I'm so sorry. Deoxycytidine triphosphate. फिर adenine को पढ़ के आएगा deoxythymidine triphosphate. Thymine को पढ़ के आएगा deoxyadenosine triphosphate. और cytosine को पढ़ के आएगा deoxyguanosine triphosphate. If you remember, who does G pair with C? Who does A pair with T? Who does T pair with A? Who does C pair with G? So all these triphosphates are delivered and new strand construction begins. But sir, ये है तो triphosphates. And actually, जब नया strand बनेगा, new strand has got nitrogen based sugar and one phosphate. तो एक ही phosphate होता है. तो deliver क्या किया गया है? Triphosphate. और बनेगा eventually क्या? Adenine nitrogen based sugar and one phosphate. तो triphosphate has to again become monophosphate. ये ट्राइफॉस्फेट है. ट्राइफॉस्फेट्स जो एक्टिवेट किए थे हमने, they will eventually become what? Triphosphate will become monophosphate. because है तो नाइट्रोजन बेस, sugar and one phosphate, and what has been delivered is triphosphate. So ट्राइफॉस्फेट का मोनोफॉस्फेट बनाना है, तो समझो दिस इज टेम्पलेट, दिस इज टेम्पलेट। उसके ऊपर है एडेनाइन, बीच में है प्राइमर मॉलिक्यूल, दिस इज ओल्ड स्ट्रैंड, बीच में है प्राइमर मॉलिक्यूल, एंड दिस इज टेम्पलेट, adenine, cytosine, guanine, thymine. And what is delivered is thymine, sugar, phosphate, and phosphate. तो डिलीवर क्या किया गया है? डीऑक्सी डीऑक्सीथाइमिडीन ट्राइफॉस्फेट, डीऑक्सीथाइमिडीन ट्राइफॉस्फेट। तो एडेनाइन के सामने डीऑक्सीथाइमिडीन ट्राइफॉस्फेट डिलीवर किया गया है। सिमिलरली साइटोसिन के सामने वी हैव गुएनोसिन, शुगर, phosphate, phosphate. So deoxyguanosine triphosphate, deoxyguanosine triphosphate. So everywhere यहां पर साइटोसिन, शुगर, phosphate, तो everywhere 3 phosphates are delivered. So why are we having 3 phosphates delivered? If you remember, ये है टेम्पलेट, ये है टेम्पलेट, ये है आरएनए प्राइमर मॉलिक्यूल, और ये है नाइट्रोजन, शुगर एंड फॉस्फेट, ट्राइफॉस्फेट। ये है नाइट्रोजन बेस, ओल्ड स्ट्रैंड, ये है टेम्पलेट, ये है बीच में आरएनए प्राइमर मॉलिक्यूल, एंड ये है गुआनिन के सामने साइटोसिन, शुगर एंड ट्राइफॉस्फेट। So what is this here? Deoxythymidine triphosphate. Then we have guanine. ये है guanine. So deoxyguanosine triphosphate. ये है cytosine. So deoxycytidine triphosphate. Why are triphosphates delivered? तो सर यहां तो सिंगल फॉस्फेट होता है डीएनए में, तो ये है टेम्पलेट, and what we have delivered is triphosphate. ये है टेम्पलेट, ये है गुआनिन, बीच में आरएनए प्राइमर, and we have sugar, and we have phosphate, triphosphate. Why were triphosphates delivered? Why was phosphorylase used for activation? Sir, ये जो दो न्यूक्लियोटाइड्स बनते हैं, 2 phosphates have in them energy which helps in removal of the water molecules. तो ट्राइफॉस्फेट क्यों है? क्योंकि फॉस्फेट के बीच में जो एनर्जी है is required for removal of water molecules, and triphosphate will become, by removal of 2 phosphates, उसकी bond energy is required for removal of water molecule. एंड वाटर मॉलिक्यूल निकल जाता है, जिससे दो फॉस्फेट की एनर्जी यूज हो जाती है एंड ओनली अ सिंगल फॉस्फेट इज अटैच्ड. So why did it get delivered as triphosphate? Because phosphate, second and third phosphate, तो फर्स्ट सेकंड थर्ड फॉस्फेट, फर्स्ट और सेकंड फॉस्फेट के बीच में एनर्जी होती है, सेकंड एंड थर्ड फॉस्फेट के बीच में एनर्जी होती है, एनर्जी बॉन्ड होता है. दैट एनर्जी हेल्प्स इन formation of a water molecule between the fifth carbon and the third carbon to form a phosphodiester bond. तो वो डिहाइड्रेशन जो होता है ना, इज एन एक्टिव प्रोसेस. इट रिक्वायर्स एनर्जी, एंड एनर्जी रिमूविंग प्रोसेस है, एनर्जी यूटिलाइजिंग प्रोसेस है. एंड फॉर दैट रीजन, टू रिमूव द water molecule and establish a phosphodiester bond, you had triphosphates which were delivered. ट्राइफॉस्फेट में से बच जाएगा मोनोफॉस्फेट, रह जाएगा एक ही फॉस्फेट. दो फॉस्फेट निकल जाते हैं, लेकिन उसके बीच में जो एनर्जी थी, उससे डिहाइड्रेशन हुआ टू फॉर्म फॉस्फोडाइस्टर बॉन्ड and the DNA strand becomes continuous. That is the reason it was delivered as triphosphates, and that is the reason first phosphorylase had activated all the nucleotides. So chalo, we have understood that, and that is not given in the book, so yaad rakhna. Who delivers the new nucleotides? DNA polymerase. Now new strand बनते जाता है. So once again, ये है template. बीच में है आरएनए प्राइमर मॉलिक्यूल, उसके ऊपर आ गया न्यू स्ट्रैंड. न्यू स्ट्रैंड इज मेड बाय डीएनए पॉलीमरेज. So new strands are getting constructed. Now we have one little problem. ये ध्यान से सुनना. The enzyme DNA polymerase moves in one specific direction. It moves from the C5 end to the C3 end of the new strand. So that is called polymerization. तो polymerization कौन से एंड से कौन से एंड में होता है? It takes place from C5 end to C3 end of new strand. तो प्रॉब्लम क्या है सर? सर, अगर पोलैरिटी C5 से C3 में नया स्ट्रैंड बनता है, तो टेम्पलेट की पोलैरिटी क्या होनी चाहिए? What should be polarity of template? If you remember, both strands are anti-parallel in nature. ये किया था हमने. Both strands are anti-parallel in nature. So, if new strand is formed in the 5-3 polarity, the template should have 3-5 polarity. तो बराबर है. Template has got 3-5 polarity, then new strand will have 5-3 polarity. ये बराबर हो जाएगा. And नया strand बनेगा in the 5-3 polarity. But sir, there is a little problem. The problem is, when the two strands are separated, one template has got 3-5 polarity, but the other template was anti-parallel to it. So it had got 5-3 polarity or 3-5 polarity, but in the downward direction. So ये था आपका ascending strand, ये है 3-5 polarity. but ये आपका जो descending strand है has got 3-5 polarity in the downward direction. And रेप्लिकेशन फॉर्क जब बनेगा, रेप्लिकेशन फॉर्क बनेगा, न्यू स्ट्रैंड इज ऑलवेज फॉर्म फ्रॉम 5 एंड टू 3 एंड। न्यू स्ट्रैंड इज ऑलवेज फॉर्म फ्रॉम 5 एंड टू 3 एंड। तो यहां ये वाले स्ट्रैंड के ऊपर ऑपोजिट साइड ये है 5 एंड, और यहां है 5 एंड, और यहां है 3 एंड। तो न्यू स्ट्रैंड कैन नॉट बी फॉर्म्ड फ्रॉम द 3 एंड टू द 5 एंड। 3 एंड टू 5 एंड नया स्ट्रैंड फॉर्मेशन नहीं हो सकता है। न्यू स्ट्रैंड कैनॉट फॉर्म फ्रॉम 3 टू 5। न्यू स्ट्रैंड कैन ओनली फॉर्म फ्रॉम 5 to 3. New strand with the help of DNA polymerase can always go from 5 to 3, cannot go from 3 to 5. ये पॉसिबल नहीं है। तो how will polymerization take place? DNA polymerase का golden rule है, it will always go from 5 end to 3 end. It will not go from 3 end to 5 end. So how does polymerization take place? ध्यान से सुनना, ये किसी भी बुक्स में बराबर से दिया नहीं है। ये है दोनों स्ट्रैंड। अभी ध्यान से, दिस इज 3 टेम्पलेट, दिस इज 5 टेम्पलेट, दिस इज 5 टेम्पलेट, दिस इज 3 टेम्पलेट। एंड अभी ध्यान से, न्यू स्ट्रैंड विल फॉर्म 5 end to 3 end on one strand. तो यहां 3 to 5 टेम्पलेट था, 5 to 3 टेम्पलेट पे न्यूक्लियोटाइड्स बनेंगे। सो एक स्ट्रैंड पे डीएनए पॉलीमरेज की मदद से 5 end to 3 end न्यू स्ट्रैंड फॉर्म हो जाता है। ऑपोजिट स्ट्रैंड 5 to 3 टेम्पलेट, DNA polymerase cannot form new strand from 3 to 5 direction. 3 to 5 डायरेक्शन नया स्ट्रैंड नहीं बनता है। तो क्या होगा? 3 to 5 टेम्पलेट पे न्यूक्लियोटाइड्स आएंगे, से 0 to 3000, 3000 तक न्यूक्लियोटाइड्स आए। अदर स्ट्रैंड 5 to 3 पोलैरिटी टेम्पलेट का 3 to 5 न्यू स्ट्रैंड फॉर्मेशन नहीं होगा। तो 3 to 5, 5 to 3, 5 to 3, 0 to 3000, 5 to 3, 3 to 5, ille, nil, no new strand formation will take place. Okay, 3 to 5, 5 to 3, 3000 nucleotides approximately. 5 to 3 template, 3 to 5 new strand not taking place. Then यहां कैसे बनेगा? 3 to 5 template, 5 to 3 new strand will form 0 to 3000, say 1 to 3000 nucleotides. Now, some of the DNA polymerase takes a U-turn. Some of the DNA polymerase takes a U-turn and supplies new nucleotides. Supplies new nucleotides even to the other template. But in the opposite direction. once again. Three to five, 5 to 3. New strand going upwards towards the replication fork. 1 to 3,000 nucleotides. 3,000 nucleotides. Yahan 5 to 3, 3 to 5. Nil. 3 to 5 banta hi nahi hai. After 3,000, some DNA polymerase takes a U-turn and supplies new nucleotides from 3,000 towards first. 3,000 towards first. But in which direction? If you remember, this was 5 to 3. This is 5. This is 3. So DNA polymerase is now going from 3 to 5 of the template. 5 to 3 ऊपर नहीं गया, but 3 to 5 नीचे जाएगा. So DNA polymerase goes from 3 to 5 of template, means 5 to 3 direction of the new strand. 5 to 3 direction of new strand. Once again, 3, 5 template, 5, 3 template, helicase, SSBP, RNA primer, DNA polymerase, 3 to 5 of template, 5 to 3 of new strand, 3,000 nucleotides, 5 to 3 of template, 3 to 5 of new strand does not take place. 5 to 3 of new strand, some DNA polymerase takes a U-turn, supplies new nucleotide from the 3 end to 5 end in the opposite direction. Opposite direction. So this was up, this is down, and this is in the opposite direction. And where is strand getting formed? From the 3 end to the 5 end. Strand is getting formed from 3 end to the 5 end of the template, means 5 end to 3 end of the new strand. So naya strand banta hai, but in the opposite direction. So if I show it video diagrammatically over here, check it. Check this carefully. 3 end, 5 end, 5 end, 3 end. ये है template. New strand is formed 5 end to 3 end, but in the upward direction. But in the opposite strand, new strand is formed from 5 end towards 3 end, but in the opposite direction. So this strand is formed upward, but this new strand is formed downward, opposite direction. This is one logic. Very importantly, ये बुक्स में दिया नहीं है, ध्यान से सुनना. ये है रेप्लिकेशन फॉर्क, एंड ये 3 end to 5 end, ये 5 end to 3 end, 3,000 nucleotides बने. 3,000 nucleotides, 5 end to 3 end of new strand. 5 to 3 of new में और टेम्पलेट में तो 3 to 5 तो कुछ बना ही नहीं था. 3 to 5 तो कुछ बना ही नहीं था. So this was 3,000, this was 0. Now while some of the DNA polymerase took a U-turn and supplied new nucleotides to the opposite direction. Supplied new nucleotides in the opposite direction, 3000 to 0, 3000 to 0. In the original other template, new nucleotides continued to be formed in the upward direction at the same time. At the same time. So, what do you mean by at the same time? 1 to 3000, 0. 3000 to 1 in the opposite direction. Simultaneously, 3000 to 6000. Oh, 1 to 3000, 0. U-turn. 3000 to 1, opposite direction. Same time, 3000 to 6000. Simultaneously. So, the strand is having 3000. The strand is having 0. The strand is now having 3000. But the strand at the same time is having 3000 to 6000. So, 3000, 0. 3000, 6000. And then again, 6000 say DNA polymerase takes a U-turn and supplies from 6000 to 3000. 6000 to 3000. But at the same time, 6000 to 3000, here it is 6000 to 9000. So, if you understood this, 3000, 0. 3000, 0. 3000, 6000. 6000, 3000 plus 3000, 6000, 9000. 3000, 0. 3000, 6000. 3000 plus 3000, 6000 plus 3000, 9000. तो एक स्ट्रैंड में रेप्लिकेशन हमेशा पहले होता है कंपेयर्ड टू द अदर स्ट्रैंड. Let's see this diagrammatically so you understand this properly. Check this here. This is 3 end. 3 end. 5 end. 3 end. 5 end. And check this here. 3000. 3000. Yahan 0. Now some of the DNA polymerase takes a U-turn and supplies new nucleotides from 3000 to 1. But at the same time, this is 3000 to 6000. So 3000 जब था, यहां कितना था? 0. Now simultaneously क्या हुआ है? Simultaneously 3000 2, 1. In which direction? 5 end to 3 end. That is golden rule. This was 5 end to 3 end. New strand is always formed from 5 end to 3 end. So 0 to 3000. 0. 3000 to 1. Simultaneously 3000 to 6000. Now check this here. 6000 say this is new 3000. So 6000 to 3000. But at the same time 6000 to 9000. So if you remember, around 3000 nucleotides is one gene. 3000 nucleotides is one gene. So yahan one gene from 3 end to 5 end. From 3 end to 5 end, one gene. From 5 end to 3 end, no gene. One gene. Now, one gene formed here from 5 end to 3 end. But at the same time, second gene formed here. तो यहां कितने जीन्स हो गए? फर्स्ट जीन और सेकंड जीन. और यहां कितना जीन बना है? फर्स्ट जीन. Now, this is second gene here. 6000 to 3000. Second gene. But at the same time, your second gene to third gene. So if you understood, first gene, no gene. I'll show it again. First gene, no gene. Now, U-turn. First gene, simultaneously. Second gene. Second gene, simultaneously. Third gene. So, first gene, no gene. यहां फर्स्ट जीन, यहां सेकंड जीन, यहां सेकंड जीन, यहां थर्ड जीन. So always in one strand, replication is earlier compared to the other strand. In one strand, replication is always earlier compared to the other strand. One gene, no gene. One gene, two gene. Two gene, three gene. So in one strand, replication is earlier compared to the other strand. That strand is called as the leading strand. What is that strand called as? The leading strand. And opposite strand may replication takes place a little later is called as the lagging strand. तो जिस स्ट्रैंड में रेप्लिकेशन पहले हो जाता है is called as leading strand. So समझो, this is 3 end to 5 end and this is 3 end to 5 end. This strand replication takes place earlier and is called as the leading strand. And yahan replication is always later, is called as the lagging strand. Lagging strand. Leading strand is formed earlier, lagging strand is formed later. Leading is in which template? 3 to 5 polarity. 3 to 5 polarity template में leading है. 5 to 3 polarity वाला कौन सा है? 5 to 3 polarity वाला lagging है. 5 to 3 polarity वाला lagging है. Leading is formed in the direction of replication fork. If you remember, replication fork ऐसे 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 separate हो रहा है, तो leading ऊपर जा रहा है. Leading ऊपर जा रहा है. Leading is in the direction of replication fork. In the direction. Leading is in the direction of replication fork. Lagging is formed in the opposite direction of replication fork. Lagging is formed in the opposite direction of the replication fork. Lagging is formed in the opposite direction of the replication fork. And you have leading strand. ये है leading strand. ध्यान से, leading strand is formed upwards. Leading strand is formed upwards and it is formed continuously. 1 gene, 2 gene, 3 gene. So leading strand is continuous. Leading is continuous. Leading is continuous. Whereas सबसे इंपॉर्टेंट, ये बात ध्यान से सुनना, लैगिंग स्ट्रैंड कैसे होता है? लैगिंग इज फॉर्म, याद है, 3000 to first nucleotide, then 6000 to 3000 nucleotide, then 9000 to 3 and 6000 nucleotide. तो लीडिंग बनता है कंटिन्यूअस, बट लैगिंग इज फॉर्म फर्स्ट जीन, देन सेकंड जीन, देन थर्ड जीन. सो इसके छोटे छोटे फ्रैगमेंट्स बनते हैं. 3000 to 1, 6000 to 3000, 9000 to 6000. तो ये छोटे छोटे फ्रैगमेंट्स बनते हैं एंड दीज फ्रैगमेंट्स आर कॉल्ड एज ओकाजाकी फ्रैगमेंट्स. These fragments are called as Okazaki fragments. तो once again, ये है leading strand, continuous. Leading is continuous. Lagging will be 3000 to 1, 6000 to 3000, 9000 to 6000. तो एक फ्रैगमेंट, ये दूसरा फ्रैगमेंट, ये तीसरा फ्रैगमेंट, और छोटे छोटे फ्रैगमेंट्स बनते हैं. Those fragments, leading is continuous. Lagging is in the form of small fragments. And these fragments are called as Okazaki fragments. What are the fragments called as? Okazaki fragments. And when this will finish, sir, ये है template, ये है template, और ये है leading strand. लीडिंग स्ट्रैंड तो कंटिन्यूस बनेगा, लीडिंग स्ट्रैंड कंटिन्यूस बनेगा, बट लैगिंग स्ट्रैंड जो है, इट इज फॉर्म्ड इन द फॉर्म ऑफ फ्रैगमेंट्स व्हिच आर कॉल्ड एज ओकाजाकी फ्रैगमेंट्स. तो पूरा स्ट्रैंड फ्रैगमेंटेड बनेगा, क्योंकि यहां तो छोटे छोटे फ्रैगमेंट्स है कॉल्ड ओकाजाकी फ्रैगमेंट, तो दोनों स्ट्रैंड फ्रैगमेंटेड बनेंगे. हाउ कैन यू हैव अ स्ट्रैंड इन द फॉर्म ऑफ फ्रैगमेंट्स? तो एक एंजाइम आता है कॉल्ड एज डीएनए लाइगेज एंजाइम. कौन सा एंजाइम सर? डीएनए लाइगेज एंजाइम. और लाइगेज एंजाइम आके ओकाजाकी फ्रैगमेंट्स को चिपका देता है. It sticks all the Okazaki fragments with each other. Ligase enzyme sticks all the Okazaki fragments with each other, and the entire DNA becomes continuous. So Okazaki fragments are stuck together by which enzyme? DNA ligase enzyme. Ligase enzyme will stick all the Okazaki fragments to each other. And once that is done, do we remember? यहां था टेम्पलेट, बीच में था आरएनए प्राइमर मॉलिक्यूल, बीच में था आरएनए प्राइमर मॉलिक्यूल, और फिर बन गया न्यू स्ट्रैंड, लीडिंग या लैगिंग या विचएवर. बीच में लेकिन कौन था? इफ यू रिमेंबर, बीच में था आरएनए प्राइमर मॉलिक्यूल. सो ये कौन था? आरएनए प्राइमर. ये कौन है? आरएनए प्राइमर. अब आरएनए प्राइमर की जरूरत नहीं है. सो एन एंजाइम कॉल्ड एज RNAs will come and break down RNA primer molecule. RNAs will remove the RNA primer molecule. And once RNA primer molecule is removed by RNAs, both strands come close to each other, और उनके बीच में हाइड्रोजन बॉन्ड्स बन जाते हैं. उनके बीच में हाइड्रोजन बॉन्ड्स बन जाते हैं. And the two DNA strands come close to each other. Hydrogen bonds are established between the complementary nitrogen bases, and the two DNA strands become continuous and start coiling around each other. तो coiling start हो जाता है. And look at this. One mother DNA has formed two daughter DNAs which are exactly identical to each other. They are exactly identical to each other. What was the sequence here? CG. What did old C get new G? What did old G get new C? This was TA. What did old T get new A? What did old A get new T? So CG, old C को मिला new G, old G को मिला new C. TA, old T को मिला new A, old A को मिला new T. AT, old A got new T, old T got new A. So what is our sequence now? This is CG, this is CG, CG, CG. This is TA, this is TA, TA, TA. This is AT, this is AT, AT. So what was mother? CG, TA, AT. What are daughters? CG, TA, AT. CG, TA, AT. Beautiful. Because of semi-conservative method, key, from one mother DNA, you get two daughter DNAs, each retaining one original strand. तो ये ओरिजिनल स्ट्रैंड था, ये ओरिजिनल स्ट्रैंड है, और ये ओरिजिनल स्ट्रैंड्स हो गए. So we have retained the original strand, and we have got new strand which is complementary to original strand. New strand is complementary to original strand. So both daughter DNAs will be genetically identical to each other and identical to the mother as well. Both daughter DNAs are genetically identical to each other and identical to the mother as well. Chalo, so we have understood this in a lot of detail. This is required at your level. इससे ज्यादा कोई पूछने वाला नहीं है आपको. But I'm just questioning you a little more. Is the process over or something else is missing? Sir, कुछ miss हो रहा है क्या? If you remember, this was the DNA. ये आपको सिर्फ नॉलेज के लिए. This is your lecture is over. This is only for knowledge. ये आपका DNA and we had taken origin. We have taken origin, incision, incision, incision. We have to replicate only one fragment. एक fragment replicate किया है. Now we have to replicate many other fragments because we had taken multiple incision with the help of incision enzyme. So what is the real thing? The real thing is this is the DNA. And with the help of incision enzyme, we have broken here. So check this here. After breaking, we will have two DNAs. एक है C, यहाँ था C3 end. If this is C3 end, after breaking, this will become new C5. And this was C5. So after breaking, this will become C5. So this will become C3. This was C5. So after breaking, this will be C5. So this will be C3. And this was C3. So this will become C3. This will become new C5. So after incision, C3, C3, new C5. C5, C5, new C3. C3, C3, new C5. C5, C5, you get new C3. Now, where will be leading from? Leading will be from C5 to C3 of new strand. C5 to C3 of new strand. And this is C5. And this is C3 of new strand. But C5 to C3 में opposite direction में बनेगा. So ये क्या बनेगा? Okazaki fragment. 5 to 3 polarity में opposite direction में 5 to 3 polarity में बनेगा Okazaki fragment. So C3, C5, C5, C3 continuous strand. C5, C3, C5 to C3 downward will be Okazaki fragment. But in the opposite strand, this is C3, this is C5, this is C5, this is C3. So C5 to C3 will be continuous strand. And C5 to C3 will be Okazaki fragments. तो यहां कंटिन्यूस स्ट्रैंड लेफ्ट था, यहां कंटिन्यूस स्ट्रैंड राइट में बनेगा. यहां ओकाजाकी फ्रैगमेंट्स लेफ्ट में बने थे, यहां ओकाजाकी फ्रैगमेंट्स राइट में बनेंगे. सो दोनों ऑपोजिट डायरेक्शन में चल रहा है. Here we have Okazaki fragments. Here we have Okazaki fragments. Here we have continuous strand. Here we have continuous strand. So after they separate, this will be continuous. This will be Okazaki. This will be continuous. This will be Okazaki. So Okazaki, 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 Okazaki in the opposite direction. Or who sticks the Okazaki fragment? Ligase. तो ligase 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 ligase. So actually speaking, this will be the way it will actually be. This is continuous. This is Okazaki. This is Okazaki. This is continuous. This is continuous. This is Okazaki. ऐसे बहुत सारे fragments है. And fragments में Okazaki are joined by ligase, 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 ligase. फिर opposite side again ligase, ligase. एंड इसको कंटिन्यू करेंगे तो ये होगा ligase, ligase. एंड ये होगा कंटिन्यूअस. दिस इज द अदर कंटिन्यूएशन. फिर यहां होगा ओकाजाकी ओकाजाकी ligase ligase. यहां होगा कंटिन्यूअस. ऐसे मल्टीपल फ्रैगमेंट्स थे. इफ यू रिमेंबर इंसिशन 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 इंसिशन, तो ये सब अलग अलग हो जाएंगे और बहुत सारे ligase से स्टिक होंगे और उसके बाद ligase will join all the fragments to each other. Ligase will stick all these fragments to each other and all fragments are stuck by ligase enzyme. सारे fragments को चिपकाता है ligase enzyme and you get two continuous daughter DNAs which are genetically the same. Beautiful logic called as DNA replication, which we have understood.